guess we'll get started for the day. So we're going to be continuing our discussion on expression data, and in particular getting into some of the issues on what you do computationally and statistically to actually learn from expression data. And as I mentioned last time, we'll usually be referring to gene expression data, although similar principles would apply if we're talking about protein expression data. So before we get to that, are there any remaining questions from the last class? Any issues about what we mean about expression, what kind of technologies we have for looking at it? Okay, well, then we can move on. This is an image that you should recognize from the previous class. So it's a way of visualizing a set of expression data where canonically we would say we have a set of columns representing samples. There might be different conditions to which we subject an organism, different locations, like different tissues in an organism, could be different patients in a population. In this case, they are in fact different patients, so these are samples in particular of lung tumors, looking at gene expression in lung tumor biopsies. And then we would have, across the left to right here, different probes, in this case probes on microarray, although they could be different regions in the genome or profiling by RNA sequencing. They could be different individual genes for profiling by RT-PCR, but basically we would have some kind of measurement of expression of different units of information. And this would be a visualization of that kind of data. And one of the things we should be able to recognize from this is that a data set like this has regularities within it. So it has patterns we can focus on. You can identify some regions of maybe similarity within it. But it's also very noisy, and that's going to be typically true of expression data. It's not so easy to say very cleanly whether any particular seeming pattern or region of regularity you can pick out is actually robustly present or is maybe an artifact of just random chance that we happen to be good at picking out patterns, whether or not there are patterns there. And that's really why we need good algorithms and statistics for working with this kind of information. So there are some basic questions we typically are interested in when we're looking at expression data. And one of the common things we want to be able to ask is, if we look at two different conditions or subsets of samples, are there genes that distinguish those? So are there what we would call differentially expressed genes between those subsets of samples? So if we're looking at a situation like the lung cancer instance we had on the previous slide, it might be that we have some samples of biopsies of lung tumors, and we have some biopsies of healthy lung tissue, and we're trying to ask, are there genes that show different patterns of activity in lung tumors versus healthy lung tissue? So that would be an example of this first question. In some cases, we don't know in advance what categories are interesting, and what we're trying to do are discover the interesting categories either within the genes or within the samples. So it might be that we are interested in finding subsets of genes that behave similarly to one another. So across all of our samples, the genes coordinately turn on and off together. So when one gene in the sample is on, they're all on, and one is off, they're all off. And that would be another example of a problem we're often interested in. A closely related problem is asking the similar question with respect to the samples. So are there groups of samples that behave in common across all the genes? So can you find different samples that exhibit a similar pattern of expression genome-wide? And that would be the final question here. And there are basically two tools we would use for asking these kinds of questions. The first question is something we typically address as a statistical hypothesis testing question. So can we show that there is a robust difference, or in other words, can we reject a hypothesis that there is not a difference? And that's commonly how we would identify genes that are differentially expressed between defined subsets of samples. When we're trying to discover the categories, we're not given those in advance, and we want to figure out which subsets of genes or samples or whatever are in the categories, then we would commonly use a class of algorithm called clustering. And what I want to do for most of today is go through these two classes of methods. So how you would do a statistical hypothesis test to identify differentially expressed genes between defined samples, and how you would discover meaningful categories either within genes or within samples in a data set like this. So are there any questions yet about what we're trying to do here? Okay, well let's go then to the question of identifying differentially expressed genes, we would sometimes also see referred to as marker genes. So if we're trying to identify marker genes, we would assume that we are given some categories in our data. So in the example I was mentioning before, we might be trying to discover genes that have different patterns of expression in lung cancers versus healthy lung tissue. And then it may be that category 1 are our samples of tumor biopsies, category 2 are our samples of healthy lung tissue. And then what we're trying to do is run through the genes and identify any gene that has a robustly distinct pattern of expression between those categories. So we might notice, for example, that there is maybe a set of genes around here that seem to more or less have high expression represented by magenta here in category 2, low expression in category 1. It's not true necessarily across the entire category. It's not exactly the same in each gene, but we might say, just visually looking at this, it looks like there is a module of genes that on average has a different pattern of expression in category 1 versus category 2, and that would be the sort of thing we're trying to discover here. So if we want to do this, in some sense, what we're trying to do is take a set of data points, and for any given gene, we can think of it as just a set of data points along a one-dimensional real number line. So if we're looking at a single spot on our microarray, it might be that category 1 is represented by these blue circles here, and then values to the right would represent high expression, to the left low expression. Maybe category 2 is the red X's here. And then we might be able to say, visually looking at this, it looks like there is on average a difference between where the X's fall and where the circles fall. But that's the kind of thing that can be very misleading if we don't have good statistics. So it's not necessarily obvious if these are far apart to a degree that would be implausible by chance. And that's where we need to think about what our model is and what we're actually trying to demonstrate in a rigorous probabilistic sense. In some sense, what we're really trying to do here is consider two possible models for how our data is generated. In model 1, we might assume that we are sampling our data from two distinct probability distributions. So we assume category 1 comes from one distribution. We would often assume this is a Gaussian or normal distribution. And category 2 comes from a different one. We would probably assume that these have different mean values. So on average, they're higher or lower in one version versus the other, or one category versus the other. We may assume also that they could have different variances. So potentially one of them might have a very tightly packed group of values, another a more spread out group of values. And that's one possibility we're trying to consider here. The alternative would be that it's just random chance. There's just one probability distribution, and all of our data points are drawn from that single probability distribution, and it's just chance that the red points happen to end up in a different average location than the blue points. And we're really trying to decide between those two different hypotheses. Now, there are more advanced kinds of methods that we're not going to get into in too much depth here. We'll see some examples of them a bit later in the class for other contexts. But more advanced methods where we can take two models like this and say which is the more plausible of these two models, using ideas we've seen in other contexts for considering the likelihood of a data set with respect to one model or another. But for the standard statistical approaches, we generally want to reduce this to one possible model and see if we can reject that model. And it ends up being convenient to think of model 2, the single Gaussian, as a null hypothesis for this situation, and then try to show that that is implausible given the data. So we will try to reject that null hypothesis. Any questions at this point? All right, so we've got a null hypothesis, and what we're going to need is a statistic that would let us reject that. And in particular, we would want a statistic that's going to look differently if this hypothesis is true than if this hypothesis is true. So in some sense, what we want is something that's going to capture the fact that a large mean difference between the gene expression values for the two categories would be indicative of a wide separation between those data sets, and would be indicative of the fact that the hypothesis 2 or model 2 is wrong. So we want a statistic that will capture the idea that if there is a large separation between the mean of the blue circles and the mean of the red x's, that's evidence that these were not drawn from a single distribution, would help us reject that null hypothesis. We need to be a little careful, though, because if you have values that are more tightly packed versus more spread out, that may affect how strong the evidence of the difference between the means is. So having means that are relatively far apart doesn't mean that much if there's a lot of variability within the two classes of data. So if you have a very spread out Gaussian distribution, you're going to, just by random chance, have your points pretty far spread apart, and it's fairly plausible you would get a relatively large mean difference, even if the points are drawn from the same data set. Whereas if we have a tightly packed set of values for each of these distributions, then we don't need to see such a large difference between the means to say robustly that we have a difference that is significant in rejecting the null hypothesis. So in some sense, what we want to consider is the mean relative to the variance. So the difference between the means of the categories relative to the variances or spreads within the categories. So you should recognize this intuition behind a statistic as something you've seen before. Where have we seen a statistic that uses this kind of intuition before? Well, this came up in a context that you should remember from last week. So this came up in the context of looking at QTL analysis. So QTLs, remember, are quantitative trait loci. The way we would commonly identify that a genetic variant is a quantitative trait locus, that is, there's a significant difference between, let's say, two different allele values of a SNP with respect to some measurable trait like height or weight, is that we would try to reject the hypothesis that the height or weight values from those two alleles were drawn from a common probability density. So in that case, we have this same intuition that if we're looking at a SNP and we have two alleles and we want to reject that hypothesis that a quantitative trait is associated with those alleles, what we would use is a t-test. So a t-test is the simplest way of doing this, and it captures exactly that intuition, that we are trying to look at the difference between the means of the two categories and see if that's large relative to the variances within the categories. So there are different versions of a t-test. This is kind of the most generic one you're likely to use. But basically what this is saying is that we will get a statistic that's equal to the difference between the means of the categories normalized by this approximately the square root of the variances. So this is a version that's accounting for the fact that we may have different sizes of data set for two categories and you want to put more weight on a data set that has more points to it. But basically what this is doing is taking the square root of a weighted average of the variance. Square root of variance is standard deviation. So more or less we're asking, are the means far apart relative to the standard deviation of the data set? And that gives us a statistic that should distinguish between whether we're looking at two data sets that are drawn from a single distribution versus two drawn from two distinct distributions. So does this make sense to everyone?
The size just refers to how many data points there are.
Yeah, so the size here is just the number of data points in each of the two categories. Okay, any other questions? All right, so to see how this might work, let's take a concrete example. Let's suppose we were looking at a particular gene, and we'll imagine that we measured activity of that gene in 8 different samples, and maybe our category 1 is our tumor samples, category 2 is our healthy tissue, and we get a set of expression values. As I've mentioned previously, you will usually look at expression values as a log ratio of the sample to some average control value. So you generally see numbers that are more or less in the range of -5 to +5, something like that. So maybe we get category 1 is -1.5, -3.1, -2.7, and +0.1. And category 2 is -0.2, +1, +0.5, +0.3. What we would want to do if we want to see if that particular gene has a statistically significant difference between these categories is plug into that t-test formula. So we would first of all need the mean of category 1. We would take the average of these 4 values here, so the average of minus 1.5, minus 3.1, minus 2.7, and plus 0.1. We would get an average of minus 1.2 there. We would also need the mean of category 2, so that is the average of these 4 values here. That will come out to 0.4. We would then need the variance within these categories. And I haven't given you a formula for the variance before, but if you know how to compute a mean, so that's just an average, it's relatively easy to compute a variance. So a simple formula for the variance is that it is the mean of the values squared minus the square of the mean of the values. So in other words, you would take the average of 1.5 squared, 3.1 squared, 2.7 squared, and 0.1 squared. That's the mean of C1 squared. And then we would subtract the mean of C1, that's minus 1.3 squared, and what we end up with is 3.1. So 3.1 is the variance of category 1. Is everyone with me so far on that? All right, so we would do the same thing for category 2. We take the mean of the squares of these 4 values, subtract the square of the mean of the 4 values, and then we plug that into our formula. And the only other thing we need is the sizes of the data sets. We have 4 data points in each category, so we get everything we need to plug into that formula, and we get out a number. So in this case, the number is minus 1.88. So minus 1.88 would be the value of our statistic for this particular data set. Any questions at this point?
Yes, the mean squared minus mean squared, I'm not seeing it.
So the mean squared is the mean of the squares of the value. So the average of minus 0.2 squared, 1 squared, 0.5 squared, and 0.3 squared. Whereas the square of the means is you take the mean of the 4 values, which is 0.4, and then square that. Okay, any other questions? Okay, so in the end, we get out a number for this. Now, the p-value testing for the t-test is not quite as simple as it was for the binomial test we saw a little while back. So in this case, there isn't exactly a simple formula you can plug into. And usually what you would use is some predefined library, so a piece of code that does this testing for you, or you could look it up in a table. There are more complicated ways if you really needed to do it yourself, but any modern programming language will have statistical libraries you can plug into, where you can just say, what is the size of your data set, what's your t-score, and it will spit back a p-value. So this is the p-value distribution for the t-test. It's a parameterized distribution, which means that different sizes of data sets and different numbers of points will have slightly different versions of the p-value distribution. So that's the distribution of values under the null hypothesis. But it converges pretty quickly on something that we can more or less treat as the distribution for relatively large numbers of values. And what all of this means is that we can basically take our statistic value and use this curve to figure out what the p-value of our significance test was. So if we were looking specifically to find genes that were overexpressed, so of higher expression in category 1 versus category 2, and we came out with a p-value, or excuse me, a t-score of minus 1.88, how would we use this to compute the p-value? What would you do? Well, if we're looking for an effect in a particular direction, that would mean that we're looking for a one-sided p-value. So what we're going to want to do is take our value, sits at minus 1.88 here, and then we're taking the area under the curve to one side or the other of that value. In this case, we're looking for overexpressed genes in sample 1, then we're looking for the right tail of this curve. So you take minus 1.88, and then your p-value would be all of the area to the right of that. So really, the majority of the area under the curve, would, if you add that up, will be your p-value. So is that likely to be a significant p-value? In this case, that's going to be pretty close to 1, you want the small p-value to have high significance. The p-value is definitely not evidence that that gene is overexpressed in sample 1 versus sample 2. If we were not sure which direction of effect we were looking for, then we would want a two-tailed p-value. So we would look at minus 1.88. We would consider the area to the left of that here. We would also consider the area to the right of plus 1.88 here. So basically this tail of the curve and this tail of the curve. Just by eyeballing it, you can say it's somewhere on the order of maybe 10% of the curve is under those two tails here, so less than minus 1.88 or higher than plus 1.88. So it's still not significant. It's maybe closer to significance, but probably still not significant. We would need more evidence to say that that gene is differentially expressed. Does that make sense to everyone? And so one of the reasons I want you to understand the formulas is that often if you are trying to design an experiment, understanding the formula helps you understand how to design your experiment, have a good chance of getting an effect. So let's say we try this and we don't get a significant result, but we think there really is an effect there, where we have good reason to believe it's there, and maybe we just haven't gathered good enough data to prove that. So we want to go back and redesign our experiment. So if you look at the formula, we should be able to pick out aspects of this formula that we could try to adjust by our experimental design to have a better chance of getting an effect. The difference between the means is kind of an intrinsic property of the gene. That's something we can't really change. It's known as the effect size that we're looking for, so that we're kind of stuck with. But we do have some other things we can play with here. So let's take the sizes. What would we want to do to the sizes to have a better chance of getting a significant effect? Yeah, we want the sizes to be larger. The bigger these are, the bigger the t-score is going to be. So an obvious way to redesign our experiment is to increase our sample size. For the same magnitude of effect and the same variance, a larger sample size will give you a larger t-score in absolute value, so you'll have an easier time detecting your effect. What about the variance? What would we want to do to the variance to increase our t-scores or increase the absolute value? Yeah, we'd want a smaller variance. So how could we do that? How might you change the variance of your expression measurements? One way we could often do that is by changing the technology we use to study them. Let's say this was a microarray study. What could we do differently to lower the variance of our measurements? Well, a more accurate assay is going to generally lower the variance. So if you have a lot of noise in your assay, that's going to increase the variance of your measurements. So if we use a more accurate technology, we're likely to have a better chance of finding a significant effect. So if, for example, we switched from microarrays to RNA-seq, RNA-seq tends to be a more accurate technology for measuring expression, so we would have probably a lower variance in our estimates here, and we would have a better chance of detecting a weak effect. If we really were just looking at a few samples here, then we could switch to RT-PCR or nanostring. That would be even more accurate, but would not be suitable for whole genome studies. So if we really cared about just one gene, you might go back and try to verify with RT-PCR, which would be very practical for one gene and would give you much more accurate measurements and therefore lower variance. So just be aware that understanding the statistics is often important in how you design the experiments. The goal of the experiment is often to see if you have statistically significant effects, and if you understand how the statistic works, that can feed into how you design your experiments. Any questions about any of that? Okay. So in some cases, we might have more than two categories. You should remember that there was a different but related kind of statistic we would often use in that case, called an ANOVA statistic. It's more or less the same intuition as a t-test, it's just more general, so you can use it when you have multiple categories. So the ANOVA test, we're basically looking at the variance of the means of our categories divided by the mean of the variances. So it's the same idea that you want the means of the categories spread far apart relative to the variability within each category. So to compute that, what we would say is we first find the mean value of category 1, mean of category 2, and mean of category 3. And then we would measure the variance, so how spread apart those means are. And then we would measure the variances within the individual categories and take the mean, and it's the average of those. And that ratio would tell us if we have a significant effect. If the ratio is high, that means the categories are far apart relative to the spread within them, so there's probably a significant effect. If the ratio is small, then we can't tell whether there's an effect. There we don't have statistical significance. Any questions about that? Right, so there is one important catch here, and that is that very often when we're looking at these kinds of studies, we're not looking at one gene. We're often trying to do this genome-wide, or at least on large numbers of genes. We want to ask if any gene from the 20,000 genes on our array, let's say, has a significantly different pattern of expression in our samples. So why does that create a complication here? Why is that a problem for us? What will go wrong if we just go through one gene at a time, look up the p-values of our t-tests or whatever, and take all the genes that have a significant p-value?
There's so many genes, there's a good chance that a lot of them are going to.
Yeah, so that's the problem of multiple hypothesis correction. If we're looking at lots of genes, then just by random chance, some of them are going to have small p-values. So you have to adjust for the p-values. That's the multiple hypothesis correction. In this field, that would often be done by von Fermi corrections, the same kind of correction we commonly use for looking for statistically significant SNPs or QTLs. And basically what that means is that we're trying to keep the probability of a false positive across our entire set of n genes low, where n might be 20,000 or so. And with a little mathematical transformation, we can say, in the limit of a sufficiently small p-value, what we really want is that the number of genes times the p-value on each individual test is small. So that would be approximately the probability that any of our 20,000 or whatever tests has a false positive. So basically, if you're trying to get a overall p-value of 0.01 and you have 20,000 genes, you need to ensure in each individual gene that you're testing for a p-value of 0.01 divided by 20,000. So that's going to need stronger evidence. It's going to be harder to get a small p-value, but that's what you need to avoid this multiple hypothesis correction problem. Any questions about that? Right, so that suggests another way that we might often think about redesigning our experiments if we run into problems in getting statistical significance. So if you do a genome-wide study and you test 20,000 genes and nothing comes up, but you think there really are some genes that kind of have a weak effect, how could this help you redesign your study to improve your chance of finding the significant one? What might you do differently? Well, an obvious thing is look at fewer genes. If you can lower this n here, then you don't need as strong evidence for any particular gene because you're dividing your p-values by n. Often that would be done by using prior knowledge, so maybe we have some prior reason to believe that some genes are relevant, and we can do a candidate gene study rather than looking at everything. There are more sophisticated methods where you might pre-screen for genes that have similar expression patterns and just look at genes that are genuinely distinct from one another and not look multiple times at ones you know behave similarly. But basically, anything that brings n down is going to give you a better chance of finding weak effects. So any questions before I move on from this? All right, so there are a lot of alternative methods for this. This is still an area people work on in trying to improve statistical design. T-test and ANOVA are basically the standard tools. Really, those with von Fermi correction will get you pretty far in this kind of work. But there are more advanced kind of tests that are out there that I'm not going to cover here. If you're really doing this in a serious way in your own work, you might want to learn a bit more about some of these more advanced tests specifically for gene expression So that brings us to the question of identifying similar patterns of activity in these experiments. So we may be interested in identifying what are called co-expressed genes. So we might, for example, want to discover that there is a module of genes here that has a common pattern of expression across our different samples. So all the genes in this module, more or less, have low expression in these samples, high expression in these samples. Basically, we have this pattern of common expression, and we might want to discover that pattern. Why might that be interesting? Why might we care about finding that? Yeah.
It shows that there's some kind of relationship, and this kind of complex relationships can be important in a disease.
Yeah, so if we observe that a set of genes have common patterns of activity across many different samples, there might be a hint that they're under common regulation. That would often itself be a hint that they perform some common function. So that's one reason we would often care about this. We might care about it for the statistical purpose I was just telling you about, that if we can discover these genes all behave similarly, then we know when doing our statistical tests that we don't need to test for differential expression of all of them. We can maybe pick one representative or one average of this set and use that to test for the entire set, and in that way bring down the strength of our von Ferroni correction. So there are other reasons, but those are a couple why we might care about these. This is computationally basically the same problem as looking for conditions that have common patterns. So identifying that maybe there's a set of patients or a set of tumors that have a similar pattern of expression across the genes in a sample. That's something we would often care about because it's telling us there's some common underlying pattern of activity that is shared by, let's say, these particular subsets of tumors, and that may be important in recognizing that these are tumors that will respond to a different kind of treatment than these tumors or these tumors. And similar things would, of course, apply in many other sorts of situations. If these were different experimental perturbations that you were interested in, so different drugs you apply to your samples, then we might want to know that some drugs elicit common patterns of expression, which might suggest that they're interacting with similar pathways within the organism. And, of course, there are lots of other instances of this. In some cases, we do what's called bi-clustering, so actually try to do both of those tasks and look for these modules of genes that more or less have common patterns across the genes and across the samples for a subset, but it all comes down to basically the same kind of method called clustering. So clustering is this problem of taking those kinds of data sets and finding these patterns of similarity. So finding subsets of samples within a larger set that exhibit similar behavior. So this is an example of a kind of problem from the field of machine learning. That's a term I've mentioned a few times. Basically, machine learning is a class of computer science that's focused on finding patterns in complex data sets or building models of complex data sets. There are different ways of defining that. But machine learning often comes in two different variants. It's called supervised and unsupervised. And clustering is an example of that. So supervised would generally mean that we have some prior information that's given to us about the thing we're trying to discover. So maybe knowing what categories some of our genes should be in. Unsupervised would mean that we're basically just asking the data to tell us what's there. We're assuming we don't know anything in advance about our categories. We're trying to discover them. For clustering, the methods we would need for one versus the other are not that different. And I'll just show you a couple of clustering algorithms. So there are a lot of clustering algorithms out there, but more or less they fall into two basic groups. They're called the agglomerative or the hierarchical methods. And k-means clustering is probably the most popular of the agglomerative algorithms. It's a very simple algorithm, but one that tends to do a decent job on a very broad class of data sets. And the basic idea behind it is as follows. So it's summarized in this set of basic instructions, which are what we refer to as pseudocode. So pseudocode is kind of an informal way of specifying a computer algorithm, and this would be pseudocode that describes the k-means algorithm. And the idea behind this is that we would first say that there is some number of clusters we're looking for. We'll call that k. And then we just repeatedly do two steps. We start by randomly assigning our points to the clusters, and then we repeatedly find a mean or average that represents each cluster, and then we assign each point to the cluster that is closest to its mean. So we keep going back and forth between these steps of finding the mean of each cluster and then reassigning points to clusters based on the means. And at some point that will stabilize. It will converge and nothing will change from one round to another, and then we know we're done with our k-means clustering. In reality, you often want to do this multiple times because the randomness can lead to different effects each time. You may not get exactly the same clusters. You may want to see what's robust among multiple runs of the algorithm, but more or less this is the algorithm. And we can walk through kind of visually to see how that would work. So this is a visualization of what it might look like if we had a set of points and we pretend that we just have two genes on which we're profiling them. So gene 1 would be the x-axis, gene 2 would be the y-axis, and then we can say that maybe points to the left represent samples in which gene 1 has low expression, points to the right, samples with high expression, and points to the bottom of the screen would be samples where gene 2 has low expression, to the top samples where gene 2 has high expression. So it's just a way of visualizing a set of samples in a space of gene expression measurements. And if we had 20,000 genes, it would be computationally the same thing. It would be a 20,000 dimensional plot, but more or less you can still think of it as points in a space. And what we would want to do then is first start by randomly assigning these points to clusters. If we have k equals 3, we're asking for 3 clusters. We just make up, let's say, 3 different colors, blue, red, and yellow, and we randomly assign our points to the 3 colors. This would be the unsupervised version of the problem. In the supervised version, we might know in advance that some of our points should be yellow, and some should be red, and some should be blue, or maybe we know that some of our points should not be in the same clusters as other points, or different kinds of information you might have. But more or less, we start with some position where we have colors assigned to our points. And we then want to find a mean for each color. We've got 3 yellow points. We would have a mean around here, 3 red points. The average of those would be around here. 3 blue points. The average of those would be around here. And we can represent those with colored stars here. So the blue star is in the middle of the blue points, red one in the middle of the red points, and yellow one in the middle of the yellow points. Right, so that gets us through the first step of that repeat loop. What's the next step? What do we do next? Right, so we want to reassign the points based on the cluster representatives, these mean values. So what we want to do is look at each point and ask which star is closest to that point. This point is closest to the yellow star, so closer to yellow than it is to red or blue, so that would stay yellow. This one is also closest to the yellow star, so it would stay yellow. But this one is closer to yellow than it is to blue or red, so this one is going to flip from red to yellow in the next round of the algorithm. These 3 are closest to the red star, so they'll become red. These 3 are closest to the blue star, so they'll become blue. So that would be the next step in our algorithm. We then go back to the first step of the repeat loop and find a mean for each cluster. That's going to shift our stars a bit, so they end up in the middle of these point sets. And then we would try to reassign our points to clusters, and we might notice that at this point it's already converged. So each of our points is closest to the star of the same color, so if we reassign our colors, they're just going to stay where they are, and nothing's going to change. So that's going to tell us that we're done with our k-means algorithm, and we now have a clustering. These 3 points are assigned to the yellow cluster, these to the blue, and these to the red. So that's basically all there is to the k-means algorithm. Any questions about any of that? Okay, so there are a lot of variants on this. As I mentioned, there are a lot of different clustering algorithms. There are different ways of defining what the mean of a cluster is, and you can get slightly different algorithms by saying that the mean is the arithmetic average or the geometric mean, or saying that it is the point closest to the center of the cluster, or whatever. There are lots of variants. And each of them is going to do more or less the same thing, but sometimes there are reasons to favor one over another. some cases, you might have different ways of measuring distance between, and there's some that have maybe more theoretical basis to them. So, for example, there's an approach called EM clustering that has a somewhat sounder probabilistic basis, but works more or less the same way, or at least at high levels, pretty similar. There are more complex methods. There are methods for automating the selection of K, so that we could figure out that we are looking at a data set where K equals 3 makes sense instead of 4 clusters and 10 clusters. But basically, what we've seen here is the K-means algorithm, and it's usually kind of the simplest default thing to try if you're trying to discover subsets in your data. Any questions about any of that? So as I mentioned, the major alternative to these algorithms are hierarchical algorithms. And hierarchical algorithms normally work by taking your data set and trying to collect points one at a time into a smaller and smaller number of subsets, and then often at the end go back and break down that collection at some level of resolution that we can decide after seeing how they've agglomerated. So what we might want to do is take the algorithm as described here, just like with k-means, there are different variations, but essentially what we're doing is more or less the following 2 steps. Repeatedly try to find your 2 closest or most similar points. Join them together and replace them with an average point representing those 2 points, or at later steps in the algorithm, maybe representing a large set of points we put together. So we replace those with, let's say, their mean. And then we throw that mean back in as if it were a point in our data set in place of the 2 we've removed. We just keep doing that until we're down to a single point. At that point, we're going to have a tree that tells us how we collected these points. Each time we join 2 points, we are putting a virtual point above them as a parent in a tree. So at the end, we've got everything down to 1 point. That's the root of our tree. And then we can simply cut the tree at some arbitrary level to give us a clustering assignment of our points. And I think that also is somewhat easier to understand visually. So I'll just go back to our example here of this cloud of points profiled on 2 teams and walk through what this hierarchical clustering might look like. So in this case, what we would want to do is find our 2 closest points. Let's say it's these 2 points here. What we're going to want to do is pull those 2 points out of our data set and replace them with the mean of those 2 points. So I'm representing that by putting this dashed circle here that would represent that there is now a subcluster of those 2 points, and that subcluster has a representative point that will stand in for the cluster in the remaining steps of the algorithm. We then do that again. We look for the 2 closest points. In this case, maybe it's these 2. We replace them with a subcluster that's represented by its mean, and we could do the same thing with these 2. Eventually, we're going to get to the point that some of the pairs of points we're joining will themselves be joining clusters of points. So maybe we join these 2. We've now got a cluster that has 3 points, which would represent this point being joined to this cluster through this representative mean point. We just keep going. We will eventually end up joining everything until we get to one big cluster with 1 point representing the entire cluster and the average of everything. And at that point, we can say that we can represent the entire thing by a tree. So this one mean point would be the root of our tree. The 2 subclusters it joined together would represent the 2 branches of the tree. And then the means of those 2 branches would be these nodes within the tree at these 2 different points. We could drill down further, kind of reversing the steps we just did, and figure out what's going on with each of these to reconstruct a complete tree, getting us down to our individual points. So does this make sense to everyone? All right, so once we've done that, we now have, in a sense, a family of different possible clusterings. Because anywhere we cut this tree, so anywhere we put a slice through it, it's going to imply a clustering of our points. If we cut it here, then we're breaking a subset of the edges. We're separating off these 2 points into a red cluster, 1 point into a blue cluster, 3 into a yellow cluster, and 3 into a green cluster. So that would give us a clustering of 4 distinct clusters of our points, and we can see how those are assigned in our original point set. But we could have cut it lower in the tree and gotten more clusters. We could have cut it higher in the tree and gotten fewer clusters. So in some cases, what we want to do is really get the tree, and we want to recognize that there are different levels of resolution at which the clustering is meaningful. We really want to preserve the entire tree to understand that some points are more similar than other points. In some cases, we would use the tree to figure out the right level to do the cutting. So we might, for example, have some certain magnitude of distance that's particularly interesting to us, that maybe we consider a significant amount of separation, and then we would just go down the tree to that significant amount, cut it, and let the data kind of tell us how many clusters should be emerging from that. But more or less, that's the idea behind this kind of hierarchical clustering, that we build this tree of similarities of points, cut it, and then we get a cluster. Now, you should recognize that at a certain level of abstraction, the algorithm I've described here is very similar to an algorithm we've seen before in a completely different context. Can anyone think of what that is? Where have we seen this mathematical process before?
Is it like the graphs for genes or something? If you have the best way to match that.
Well, that does also use some graph theory. So they're both kind of graph problems in a way. There's something that's even closer to this. And that was back towards the beginning of the class when we looked at sequence alignment. So we had seen an algorithm for multiple sequence alignment, the plus LW algorithm. So plus LW is basically the same thing as what I just described here. So in the cluster LW algorithm, remember, we are multiply aligning a set of sequences, and the way we do this is we repeatedly look for the two most similar sequences in our set, we align those two, and then we replace them with an average sequence, so a consensus sequence, remove the two original sequences from our data set, throw in the consensus, and then repeat that until we get back to a single consensus. There, again, we were ending up with a tree, where in that case, the nodes of the tree would be labeled with sequences, so putative ancestors of our leaf node sequences, rather than kind of average expression values. But at the right level of abstraction, it's basically the same process. It's kind of a generic progressive algorithm for solving a hard computational problem by turning it into the process of repeatedly solving an easy computational problem. So I point that out because I think that's one of the things that goes into learning to think like a computational biologist, that a lot of what you're trying to do is look for these similarities. Look for cases where maybe very different biological domains can be brought back to similar mathematical abstractions, and then by understanding how people solve that abstraction in a different context, you can learn ways that you can help solve it in a new context. So that's just something to be aware of, that this particular kind of hierarchical algorithm comes up other places, may be a useful trick in other kinds of contexts. So just like with the k-means, there are other variants on these kinds of hierarchical algorithms. There are different ways of defining what the average of a cluster is, or the mean, or the median, or geometric mean, or arithmetic mean, various ways of doing that. Different ways of measuring distance to decide which things are closest together, or how far apart one cluster is from another cluster. And there are even versions where you do this top-down instead of bottom-up. That's computationally a lot messier, so it's not done as often. But just as with the k-means, there are lots of clustering algorithms out there. But if you understand the k-means and the hierarchical method I told you here, I think you understand more or less how clustering works, and you've got a couple of techniques that between them will be appropriate for almost any problem you're likely to encounter. At least will do a decent job for almost any problem. All right, so we're about out of time here. Are there any questions about any of this before we break? Okay, I guess I'll see you Friday when we get into actually looking at some of these expression data.